Hello, everyone, and thanks for the invitation, and thanks for having me. So yeah, like Celia mentioned, I'm gonna talk to you about extremophile hotspots linked to containerized industrial waste dumping in the deep sea basin. And I'm a part of Paul Jensen's lab at Scripps Oceanography in California right now. Let's see. Okay. So the story started with the discovery of a deep sea waste dump site in the San Pedro Basin, which is located between Los Angeles in California and Santa Catalina Island. That made headlines in 2021, where scientists discovered all these barrels on the seafloor that were possibly laced with DDT, and those newspaper articles were based on two major publications by the group of Dave Valentine and Santa Barbara that initially discovered those barrels with an ROV, so an underwater operated, remotely operated vehicle. And then another wide area survey of this whole area that identified over 70,000 targets, so objects on the seafloor that were, we didn't really know that were there. So the hypothesis and assumptions at the time was that those barrels contained acid sludge waste with DDT, which is this chemical here on the right, heavily chlorinated, and they're surrounded by microbial mats, like those white rings that I just showed you, and had some concrete footings for ballast. And so DDT was a very popular insecticide back in the days. And there's a lot of history of this chemical in Southern California because Montrose Chemical Corporation was the largest producer of DDT and was located in Torrance in Los Angeles area. And they discarded a lot of their chemical waste in the sewer system and also through ocean dumping by means of the California Salvage Company that would take whatever waste anyone would give them and ship it out to any of those designated dump sites that you can see on this historic map on the right here, and drop it in the ocean. And later on, DDT got banned because of adverse environmental effects, and it upscales through the food web, so a lot of negative impacts there. And then the Palos Verdes shelf, which is indicated in red, in this red box-ish shape, was designated as a Superfund site with a lot of money dedicated to restoration, remediation, risk communication of this pollution in this area. And then in 2011, Dave Valentine, like I mentioned, found 60 barrels in the San Pedro Basin with DDT concentrations in the sediment that were much higher than anything reported at the Palos Verdes shelf. And then this brought this whole issue back to attention of the media and also the general public, including lawmakers. So in 2022, there was finally some funding awarded to Scripps Oceanography and also through the Sea Grant program to actually study this situation and see what's going on. So we had the chance to be on the Falcor, which is from the research vessel from Schmidt Ocean Institute that had this ROV Sebastian. And we could visit that site and to do 2 dives in the area where we detected 24 barrels, and we could sample 5 of them in total. And this is some examples of how it looked like down there. So there's these corroded barrels on the ocean floor. Some of them have these white rings around them, quite ghostly looking. And yeah, and this was how we collect the sediment cores in proximity to those barrels. And our idea was to take samples in a gradient away from those barrels, like really close, and then in a distance, with the assumption that if those barrels are full of DDT, we would expect less pollution further away. But what we found is that for barrels that had this white ring around it that we refer to as halo, we could not get any sediment cores really close to the barrel. So the closest we could get is through this white halo, like about 1 meter away. And that between the halo and the barrel, there was like some hard concretion underneath. And so from one barrel, we could also take a sample of this hard mineral layer. And then this is an action shot of us on the ship processing all those cores and asking those two questions. Majorly, what are the effects of this DDT in the sediment on the microbial communities? And can we potentially identify microbes that would remediate this contamination? So for that, we first had to get DDX concentrations. So DDX is we refer to as DDT and its major breakdown products that are on the upper right here. And what we found is that this DDX, so all the different breakdown products, including DDT, are widely distributed on the area. There is no trend of decreasing concentrations further away from those barrels. But we also found that the highest concentrations were between 2 and 6 centimeters down in the sediment, so somewhat buried. And then we looked at the microbial communities. And so for that, I extracted microbial DNA and then did microbial community profiling using 16S rRNA marker genes. And while extracting DNA, I noticed that from those samples, from those halos, it was very difficult to get anything out of there. Some samples almost had no DNA in them. And troubleshooting that, we realized at some point by measuring the pH that those samples were extremely high. So we had pH of up to 12 in some of those, in those halo samples, whereas the pH in all other samples was neutral. And then we got microbial community data where we also saw that microbial diversity was really reduced in those halo samples, where in all other samples up here, we had a very diverse, very even community, kind of typical for sediments. Here I show you only the 50 most abundant taxa, so it's extremely diverse. But when we looked at those halo samples, those communities were dominated by only few dominant microbes that were all from the Bacillota phylum. Most of those were Clostridia. And then when we look at the relatedness of those dominant taxa to other database entries, we found that most of them were really most closely related to alkalophiles, so microbes that are adapted to life at high pH. That can be found, for example, in the deep subsurface, at the Lost City hydrothermal field, which is this hyperalkaline hydrothermal spring on the mid-Atlantic ridge. Others had relatives from mud volcanoes or from deep sea drilling samples, which is again the deep subsurface. And then, yeah, that really showed us how pH drives microbial communities composition in those halo samples as compared to all other samples. Where we did look for DDX, there's not much of an effect there. The concentrations are probably too low to have a pronounced effect on the community composition. It's somewhat related to sediment depth, which is a much stronger driver of microbial diversity in the area. But pH really stood out as driving those halo samples in a very different direction. And then when we looked at how the communities are related to other hyperalkaline environments, we found that samples from those halos were much more closely related to, for example, hyperalkaline spring samples in Italy than they were to their 1 meter, 2 meters away neighbors in the San Pedro Basin. And yeah, so that really showed us how those are alkaline hotspots of microbes that live there. And then metagenomics also showed us some more details on the actual adaptations that those microbes have. So if, for example, the frequency of sodium antiporters, sodium-driven ATPases, and sodium symporters, for example, were much higher in those halo samples as opposed to control samples. And it could also confirm that the main inhabitant in those halo sediments is from the genus Alkalophilus, whose closest relative is from a sub-seafloor crustal aquifer on the mid-Atlantic ridge. So again, the deep subsurface, which is really highly alkaline as well. And then the last piece of the puzzle was this concretion. So our colleague Kira at the USGS analyzed the mineral composition of this crust and determined it was mostly brucite, which is only common at high pH hydrothermal environments such as the Lost City hydrothermal field. And then I went through the chemistry of cement and concrete literature, which is not my usual literature, but anyways, it said that yes, you can make brucite from seawater if you add an alkali that brings the pH above the precipitation point of brucite, which is 10.5. And so Kira also measured the mineral composition in those sediments of halo sediments and normal sediments, and determined that the main contributor of minerals in the halo is calcite and not mica like it is everywhere else. And so with all of these things together, we, as the LED Bible recently put it, we finally solved the mystery of eerie hail barrels submerged off the US coast. And so what we think happened is that caustic waste was containerized and was dumped in the San Pedro Basin back in the days. And the acid waste that was the main source of waste from DDT production was bulk dumped, just put straight into the water, explaining this wide spread of this contamination. But when this caustic alkaline waste reached the bottom, it all leaked out because all those barrels were broken, otherwise they wouldn't sink, right? So this caustic waste seeped down into the sediment where it precipitated this brucite hard mineral layer. And then as this hard layer dissolves slowly over time, it releases hydroxide, and where these interact with the seawater, we get this constant precipitation of calcium carbonate from seawater, which would explain how these white halos form in this shape around those barrels, and also how we see them since they're constantly reformed and not really covered by sedimentation. And so this high pH under those halos then selects for a specifically adapted low diversity microbial community that has very specific adaptations to live in those environments. And that is very different from the generally high microbial diversity that you find everywhere else. And so with that, I just wanted to summarize that DDT does remain a pollutant of concern in the Southern California ecosystem, but the levels have decreased over time and the majority is buried in sediment by now, so it's a little bit out of the way. But those other barrels created extremophile hotspots that will outlast us for millennia based on the dissolution rates of this brucite mineral that we estimated. And we don't know what the total extent of this problem is. Like I mentioned, we have 5 that we sampled. We observed like 24. We don't know how many are out there in total. And those barrels are not necessarily linked to DDT production. There was also alkaline waste produced, but they could also be derived from a variety of other industries that were dumping waste out in the ocean at the time. And what else was in those barrels, we don't know. And so we still haven't answered our question on how microbes can break down DDT. There is still a lot of detectable in the environment, and so in the future we're thinking about how to help stimulate this microbial breakdown to hopefully fix this a little bit. And if you want to know more about this whole project, you can look at the QR code on the right. There's more research and more people involved in this. But for now, I would like to just acknowledge my lab and all my collaborators, as well as funding sources, and you all for your attention.